Great, thank you very much, Herman, and thank you for coming, everybody. As Herman said, I'm going to talk about genomics today, so how you can do genomic analysis and how people are doing it today using Apache Spark. At the moment, I'm a data scientist at Cloudera, and I have been for a few years. Before that, as Herman said, I worked on Hadoop itself, so working on, as an engineer, on MapReduce and Yarn and HDFS. And I'm probably best known for being the author of Hadoop, the Definitive Guide. So before I dive into some of the Spark-related things, I wanted to give a kind of brief intro to genomics in a very basic way, and that's because I'm not a biologist, so I don't understand all of the intricacies, but there are a few kind of terminological things that I want to kind of run through. What we study in genomics is individuals, and actually populations of individuals are very relevant. And so the organism we study is the human, and the human is made up of trillions of cells, and each cell contains a copy of the genome. So it contains a copy of DNA, and it's roughly correct to say that every cell has the same copy of DNA. So that's not entirely true, but it's good enough for this talk. And the picture on the right there is all of the chromosomes in a human cell. There are 23 pairs, and you can think of those as being linear sequences of base pairs with letters A, T, G, and C, which you've probably heard about all of this, but that's a kind of brief summary. So the simplification that we have here is that you can consider each individual's genome as this linear string of base pairs. And I've kind of shoved them all onto one chromosome as a simplification. You can kind of line the chromosomes up end to end, and then you've got a 1D coordinate system, basically. By the way, this clock is not advancing, so I have no idea how much time I'm using. So what we want to do when we're analyzing large populations of individuals is we want to understand the difference between individuals. And the way that we do that is by introducing an idea of a reference chromosome, or a reference genome, more accurately. And where does that come from? Well, you've probably heard of the Human Genome Project, which published its first draft at the beginning of the century and has since been refined multiple times. So in the intervening years, multiple copies of the new versions of the genome have been published. And it's a bit like software engineering, where we release a new version of software. There's a new version of the reference genome, which is a version. So we're on version 38 or so at the moment. Anyway, so what we do, because humans differ in a very small way, at a very small number of places in the overall genome, it makes sense to do a diff, as it were, between the reference and yourself. So you can look at a single location and look at the differences at that location. And this is a typical sequence of steps from getting from a sample from a person and getting to some kind of information that we can store on a computer. So you extract some DNA, you put it in a tube, you put it in this big machine for a couple of days or something, and it will spit out these ASCII documents which have reads in them. And the idea is that you can reconstruct the genome or information about the human organism by doing this magic thing called bioinformatics. So I just mentioned reads. Reads are these little snippets of DNA sequence, and in some sense they're not really a natural thing. They're a kind of artifact of DNA sequencing, because there are chemicals that chop DNA up into little pieces, and then the challenge is to recombine them to reconstruct the genome. So in software engineering parlance, it's kind of a read is like an implementation detail. It's a way of getting back to the genome. And there are other sequencing technologies that don't have this kind of short reads. Variants, on the other hand, this is another term that comes up all the time. Variants are parts of the genome where individuals' DNA differs. So I might have a G in one position on my genome, and someone else might have a T, for example. And the collection of those variants is stored in a file called a VCF file, which stands for variant call format. And in kind of computer science terms, you might think of that as like Unix diff between yourself and the reference genome. So variants are what we're trying to get, because that's what research scientists are interested in. They want variants for a population, and then they can start looking at the different variants and try and understand the association between disease and those variants. So the pipeline that we have can be divided into two parts, upstream and downstream, and I'm going to talk about both of them, and I'm going to talk about the Spark technology that implements that part of the pipeline. So upstream is where we have, like on that diagram earlier, we have the DNA samples, and we go all the way through to producing this file of variants called the VCF file. And that's done by a program in Spark, which is called GATK, which I'll talk about next. And then the downstream part is where the scientists kind of live, and they want to do analyses on collections of variants using this program called Hale, which I'll talk about in a bit more detail. So before I move on to that, actually, I'm going to talk about why this is relevant now and why genomics is such a big deal. Since the Human Genome Project at the beginning of the century, the cost per genome has massively decreased. So in 2001, this chart says that it was $100 million to sequence a genome. That's when they were doing like 1 or 2. That was the reference genome. And then over time, we've come down to sub $1,000 cost for doing the sequencing. And that's beating Moore's law by a massive multiply. And so this kind of economic effect has spurred lots of different organizations into doing very large, ambitious genomic projects. This is one. This is called the 100,000 Genomes Project, which is being run by the NHS in the UK. And as the name suggests, the project will sequence 100,000 genomes from almost that many people. And so that's been enabled by the technology, that's the sequencing technology. And if we kind of extrapolate that forward, we find that it's just going to grow more and more rapidly. There are various estimates. We're on around maybe under a million genomes at the moment. But over the next 10 years, that's going to increase to, well, estimates range from 60 million, which is on the low side, up to 1 billion, which is a huge number, obviously. And even more than that, the size of genomic and omic data, which is more than just the genome, is going to be even bigger than that. There's a very interesting paper called Big Data, Astronomical or Genomical, which looks at 4 different data sets and is trying to predict which one's going to grow the fastest over the next 10 years. So it's astronomy data, genomic data, YouTube, and Twitter. The last 2 are very low in kind of growth. Astronomy is the next, is number 2, and then they think that genomics is going to be the biggest growth. And it's because it's not just the DNA data that I've just been talking about. As I said at the beginning, DNA data, you have 1 copy in every cell, and it's basically the same. So, you know, if you sequence all the humans on Earth, that's, you know, whatever, 7 billion. But that's a bigger underestimate because you can sequence other things like RNA, which is in the cells, and you have multiple RNAs in a cell. They vary over time. There are different cells. So you can just see how this thing multiplies. And the challenge is going to be, can the technologies that we have keep up with the growth of sequencing? like, will Spark scale, basically? There's a challenge for us. Okay, so now I'm going to talk about the upstream pipeline, and this is going from the DNA sequence to the variants using a tool called GATK, which stands for the Genomic Analysis Toolkit. And GATK is a very popular piece of software. It's existed for a long time now. It's used in lots of institutes across the world. It's developed by the Broad Institute in Cambridge, Massachusetts. And so the first 3 versions are run on a single machine. They don't scale. They don't run on Spark. And so over the last 2 years, we've been working on a version that will run on Spark and will scale. And so I'm just going to talk about some of the challenges and some of the techniques we've used there to make it run on Spark. As you might imagine, like many pieces of software, it's basically a pipeline system. So you start, you put some data in at the beginning, and it runs through these different steps, and then it spits out at the end something that's useful, that's been transformed, and we get variants out of this pipeline. So the first 2 steps are alignment and deduplication, and I'll go into a bit more detail on those now. So alignment is, so when you do DNA sequencing, I mentioned that you get these reads, and the reads are just little strings, and we have no idea where they fit in the genome, whether, what position they come from. So they're unaligned. So the process here is to find where they match the reference genome. But it's a little bit more complicated than that because we're not looking for exact string matches. Because we have variations, the bases can differ from the reference. So we have to do fuzzy string matching. So that's a kind of very interesting area in itself, and I'm not going to talk about that anymore. But the interesting thing, I think, is the number of aligners that people have written over the years. And with a big peak in 2009, and well, between 2009 and 2013, lots of people wrote aligners. And there's a kind of long tail effect here. People use, there are a few aligners that are very popular, and everyone uses those, and then there are lots of aligners that aren't very popular at all, and no one uses them, but it was an interesting research project. The aligner that I've been involved, that people use that I've been involved with is BWA-MAM, and it's based on the Burrows-Wheeler transform, which, if you haven't heard of it, you may have used it actually, because it's the same mathematics as the compression algorithm in BZIP2. So that's quite an interesting thing in its own right. But the key point here is that alignment is embarrassingly parallel. You can take a bunch of reads and you can do alignment on those, and you don't have to communicate with another task that's doing alignment on another bunch of reads. So what we did is we wrote a Spark program that used BWA-MEM, which is a C program, to run BWA-MEM in parallel on a cluster. And we actually used JNI to call out this C program. And the reason we did that rather than rewriting it in Scala or Java, which would be tempting, is because the bioinformatics community is pretty conservative, and they want to be absolutely sure that the code you're running for alignment is the code that they know and love in their C library. So it was a kind of pragmatic decision to do that. And this diagram shows the idea and what's happening on the cluster. On the left, we have unaligned reads. So we've got 6 reads in this diagram, and you can imagine they've been partitioned into 2 partitions, 3 in each partition. The first 3 go into one task, and the BWA program will run and have a local copy of the reference so that it can do alignment, and then the other partition at the bottom there is processed by a separate task at the same time, and then they're kind of just joined again. This actually feeds into the next step in the algorithm, so this is the embarrassingly parallel part. So fairly trivial thing to do, actually. I'm not going to go through all the steps. I'm just going to show another step now, which isn't embarrassingly parallel. It's called mark duplicates. And the idea here is that when you do sequencing, you have, it's a very noisy process because there's lots of kind of chemistry happening. And the DNA fragments that have been created are often sequenced multiple times, and you have to remove the duplicates so that you don't have biases later on in the pipeline. And if you think about what deduplication is in general, the way you would implement it in Spark is typically you would do a sort on your data set by the key that you want to deduplicate by. And then because you've sorted, you can just run through the data set and remove duplicates that are sitting next to each other. And this is where Spark has proved to be really very useful and very powerful, because Spark has an inbuilt sort algorithm. We can just use it and build on the work that the Spark engineers have done over the years to tune that algorithm. In contrast, the previous version of GATK, GATK3, has a custom on-disk sorting algorithm that was written by the GATK developers, and it works on a single node, but it's not scalable. So there was no way for them to take that and scale it up in any way, in any kind of easy, approachable way. This is a diagram which shows the process. On the left, we've got two fragments. The colored circles are base pairs in DNA. And you can see that fragment 1 has been turned into 3 reads because of this kind of noisy process. And fragment 2 has been turned into 2 reads. So we do a sort, and then we remove the duplicates that we find after sorting. So a pretty simple algorithm, but something that Spark has really helped with. And the kind of takeaway is that performance is a lot better. This is a different tool in the pipeline, but it illustrates the point. On the left, we have the total CPU time to do the computation. And the left bar in the left diagram is a single node, a single process doing that work. And then on the Spark time is this one. You can see that it's slightly higher than the single process because there is some overhead, obviously, in running things on a Spark cluster. But it's not, particularly significant. And obviously, the saving is in the wall clock time, which is the diagram on the right, where you see that it takes a long time with one process, whereas Spark will allow you to do computations in minutes rather than hours. So if we go back to the pipeline, I talked about the first 2 stages in the pipeline, and those were relatively simple Spark jobs. The rest are a little more complicated, but they fit very nicely with the Spark RDD architecture and the transformations that Spark provides. And the other nice thing is that we can chain all of these steps together, and we can run them without having to materialize any of the intermediate data to disk, which in the previous version of the pipeline, it was a very file-intensive process because each stage would have to write its results out to disk, and we get savings from that as well. So it's nice to be able to scale up existing tools, but one of the even better, I think, is when you can do things that previously weren't possible. And while we were working on the porting of this tool to Spark, another group at the Broad were able to write a program that did something that they'd always wanted to do but could never run because the runtime was too expensive. So they did some work on structural variants, and when they implemented this in Spark, they said that it was running at what they estimated was 1 or 2 orders of magnitude faster than if they'd done it using the older serial version. So that's a really nice effect when that happens, that you can make new things possible. So that's the GATK. So we've got some variants now, and now the scientists come along and they want to do analysis of populations variants, and that's using a tool called Hale. Hale is this was written using Spark from the outset, and it's an open source tool developed by the Broad Institute again. So the idea here is that you are looking for genetic trait associations. So if you have populations of controls and cases. So that the cases have some particular disease typically. So for example, type 2 diabetes, and then the controls don't have that disease. And then you're looking for associations between the genetic variation and the disease. Most diseases are actually polygenetic, so that means that there are multiple genes involved. You've probably heard of monogenetic diseases like cystic fibrosis or Huntington's disease, which are caused by a single gene. It's much harder, obviously, if you've got multiple genes influencing a trait or a disease. And so the challenge is to be able to do this analysis across a very large set of individuals, maybe 100,000, like the 100,000 Genomes Project, or 1 million, across the whole genome. So you're looking at all of the variant sites in the genome. And the way that Hale approaches this is by having this huge matrix, and this is the heart of Hale. So if you understand the data structure, the operations that it's doing on it are relatively straightforward and easy to understand. So we have, across the top, we have samples. So each sample is a DNA sample from an individual. And then down the rows, we have variants. So each position is a known variant in the genome. So this is tens of millions of rows with up to 1 million samples, maybe. And then the point, the cell where these two intersect is what that variant was for that individual sample. And that will tell you the letter at that base, typically. And that's called a genotype. So you can use Spark to analyze this large matrix. And the way that the matrix is stored is in Parquet. So the underlying file format is Parquet. It's not actually using one column per sample because we had thousands, or maybe up to 1 million samples, and that isn't a structure that Parquet will support. So it has to have a custom encoding to allow that to be stored properly in Parquet. And the way that Hale works is it operates using RDDs. So you can read and write these data structures using RDDs. You can also use SQL engines like Impala to query it as well because it's stored in Parquet. So it's kind of accessible to other tools in the Spark and Hadoop ecosystems. So if you've got your matrix, you want to typically do an experiment, and that involves joining it with other data sets. And this is not Hale. This is a genome browser, which is a way of looking at the chromosome, which is up here. We're looking at this little section of it, indicated by this red mark. And then down the side, we've got all this other information. These are called tracks, and they have information from biological databases, which are accessible to scientists. And they store various things. For example, they might store gene information or comparison with other organisms like elephants and mice and things. And so what Hale does is allow you to join databases like that with the whole of your data set. One example is ENCODE, which I've mentioned here, which is the encyclopedia of DNA elements, and that has information about all the genes, where they occur in the genome, and positions of regulatory parts that control the expression of those genes. So Hale makes it very easy to join those databases with a VDS so that you can do analysis. You can also have annotations on samples, which you would do to say, this sample has this disease and this sample doesn't. So it allows you to have your control and case annotations in there as well. So I'm not going to dive into the details of Hale, but I'm going to just show you this notebook, which is a way of interacting with Hale. So Hale exposes a Python API, which is very accessible to scientists. It has lots of built-in functions. So here we're doing a linear regression between variants and one of the database information. And I think this shows that there's no association. But this is the style of working. You use a Python API to run your analyses. Okay, just to finish, Hale's already being used on to generate and to do QC and processing on biological databases that are being released currently. The genome aggregation database was released this year, and Hale was used for some of the processing in that. It was used to process over 100,000 exome sequences, so not the full genome, but part of the genome, and over 15,000 whole genome sequences. And then there's another one called the UK Biobank, which has a huge amount of data on UK patients, samples, and it was used to analyze it, generate some statistics for almost 500,000 individuals. And I haven't got the sizes of clusters here, but I think they're in the hundreds. So I think what I wanted to show was that Spark is being used for genomic analysis today. It's a great fit. Going back to the projections from the beginning, it's going to need to scale more over time. And my message would be, get involved in some of these projects are really, really interesting, and they need people who know about the scaling, the computer science, and there are some links there with the projects, the open source projects. And I'd like to say thanks to all the collaborators on those projects from the Broad, but also external collaborators too. Thank you very much. I think we've got time for questions.
Thank you, Tom. I think we have about 2 or 3 minutes if there are any questions. If you can step up to the mic in the middle, or I can bring this to you if you're up front.
Is my understanding correct that the entire pipeline is almost, let's say, the right side that you showed entirely Spark-based, but then the first step you say this is really a piece of tool that is not in Spark, because you say it's extremely well for the parallelization, but it's really so the alignment part. Yeah. So how can you elaborate a bit? How do you because you don't use the parallelism of Spark at that moment, so how do you parallelize that in your pipeline?
Yeah, so you actually do use the parallelism of Spark. They're running on different tasks, different executors in Spark, so you are benefiting from parallelization. I mean, but it's a good point. You don't have to run that part of the pipeline on Spark if you don't want to. Lots of institutes already have that running efficiently, so you can start this pipeline at the dedupe stage.
Hi. I wanted to ask how this work relates to the ADAM project.
That's a great question, yeah. I didn't mention ADAM. I should have listed some of the other projects in this space. ADAM is one of them. ADAM is the equivalent of the GATK part, so this pipeline. And it's an independent project. It has a few differences. ADAM also defines formats for some of these data sets, like the variant data set that not the Hale thing, the VCF data set. It defines some file formats for those that are based on Parquet, which are more efficient. But in some ways, if you look at it as a black box, it's doing a very similar thing to this. So I think ADAM's great because it's another implementation of the same thing, so it provides a comparison or another way of doing something similar. So there's some slight competition between the two, which is a good thing, I think.
Hi, thanks for the talk, very interesting. You mentioned that on alignment phase you're using this external C library, which is like it's proven and it works very well. And you mentioned that alignment is also fuzzy matching. So I assume it's like probabilistical matching, isn't it? Like it's not exact matching. So given that, how is it that it's still embarrassing parallel? Like, why is it others not influencing the probabilities of the matches?
That's a good question. I'm not sure I know the answer, but I think, so I'm not sure it's entirely probabilistic. I think you say you can tolerate a certain number of differences, and that is deterministic. So I think that's why it works, but I'm not entirely sure. Yeah, I think it's not entirely probabilistic, but which is why it has the property that it is, is parallel, and you'll get the same results. But I'd need to check whether that's completely true.
Great. Well, thank you again, Tom.
Thank you.